Welcome to part 5 of this video on the hypothalamic-Pituitary-Adrenal axis. Okay, so we're currently talking about the effects of cortisol on different tissues around the body, and we've now looked at the metabolic effects of cortisol, and we've seen that it will raise fasting blood glucose level, it will also raise the amino acid concentration in the blood, and also the free fatty acid concentration within the blood. Okay, what I now want to talk about is the effects on the immune system of a rise in the level of cortisol within the bloodstream, and it's going to have two major effects: an anti-inflammatory effect and also an immunosuppressive effect. And what I want to communicate to you is what the difference between those two effects are. What do I mean by anti-inflammatory and immunosuppressive? How are those two words different? Okay, so this requires us to understand the immune system. So I'm going to have a bit of a reminder of the immune system initially, and then I'll talk about what cortisol is actually going to do to the immune system. So remember, the immune system is a system that is designed to protect you against pathogens. So if your tissues get invaded by bacteria, viruses, fungi, parasites, the immune system is a system that is designed to deal with that. Okay, now the immune system can be divided up into two major strands: the innate immune system and the adaptive immune system. So we'll divide the immune system into these two major categories: the innate immune system and the adaptive immune system. And the difference between the innate immune system and the adaptive immune system is that the innate immune system, the response that the innate immune system is all about, is exactly the same no matter what pathogen is present. Okay, so the innate immune system is a response that will be initiated the same no matter what pathogen is present. So it does not tailor the response to the pathogen, whereas the adaptive immune system, as the name suggests, it is adapted to the pathogen. It is a response that is specific to which pathogen is currently infecting you. So it will differ depending on which pathogen is actually infecting you. So we have the innate immune system and the adaptive immune system. So let me just go over briefly what the innate immune system and the adaptive immune system involve. So the innate immune system is all about the inflammatory response. I mean, people cluster other things like the mucociliary escalator and the acid in the stomach as well into the innate immune system, but we'll just talk about the inflammatory response. This is the major component of the innate immune system. Of course, the mucociliary escalator is extremely important in keeping your lungs free of pathogens, and the stomach is the low pH of the stomach is extremely important in keeping the stomach free of pathogens, but we'll focus in on talking about the inflammatory response as the major component of the innate immune system. So the inflammatory, and I think actually it should have double M there, so I apologise for that. Imagine putting another M in there. Inflammatory response. Okay, so what is the inflammatory response? Well, let me just describe this to you in brief. So, let's say we've got some tissue, a little piece of tissue, and let's say that this portion of tissue gets infected with a pathogen. So, let me just draw a little picture. We'll have it, and I'll just raise this up. We'll put the picture here. So, we'll have our tissue here, and let's say we've got some pathogen present. So, let's say we've got a bacterial cell, okay? And it might be a gram-negative rod of some sort. So, here it is. Here is our bacterial cell. And it's dividing and dividing and dividing and making a whole population of bacterial cells in that tissue. What is going to happen? Well, what's going to happen is initially there's going to be the recognition that the pathogen is actually present. Now this is done by special cells that are present in all tissues in the body, known as sentinel cells. And there are two major types of sentinel cells: macrophages and dendritic cells. And I will draw a little dendritic cell here. So dendritic cells, the reason they're called dendritic cells is they have dendrites, which are very different from the dendrites of nerve cells. These do not have the ability to conduct an action potential, okay, but they look like these star-shaped cells like so. So this is a dendritic cell, Okay, and the reason it's got these dendrites is so that it can feel all around the local area. It's got sort of hands, if you like, these processes that are stretching into all of the surrounding area, and that really fits with what this cell is actually doing. This cell is constantly on the lookout for the presence of pathogens. It's constantly on the guard and looking out for the presence of pathogens, and if it finds evidence that there are pathogens in the tissue that it's present in, it will initiate the inflammatory response. So dendritic cells are an example of what is known as a sentinel cell, these guard cells that are all the time looking for pathogens, and if they find it, they're going to initiate the inflammatory response. Another example of a sentinel cell are resident macrophages. So in all the tissues of the body you have some macrophages that are just continuously present, and these are known as, I'll just add that additional word in, resident macrophages. And these two their job is just to sit there and look for evidence of pathogens, and if they find evidence of pathogens, they're going to initiate the inflammatory response. Okay, so in all the tissues of your body you have these sentinel cells distributed all over the place, which are looking for evidence of pathogens. Now, how are they going to actually recognize the presence of pathogens? Well, what they will recognize is molecules that are associated with pathogens. You see, bacterial cells, they produce all sorts of different molecules, and some of those molecules would never ever be present in human tissue. Human cells would never ever make those molecules. Therefore, the presence of those molecules is a giveaway sign that there is a pathogen present. There is something that is not good present there. And all of these molecules that pathogens can produce, which would never ever be produced by human cells, and therefore are a giveaway sign that a pathogen is present, are clustered under the umbrella term PAMPs, which stands for Pathogen-Associated Molecular Patterns. So the first P is for pathogen, the A is then for associated, and then the MP is for molecular patterns. So pathogens can have molecules which would never ever be produced by human cells, and therefore these can be used by the sentinel cells as the giveaway sign that a pathogen is present, and these are collectively known by this umbrella term, pathogen-associated molecular patterns. So what will happen is the sentinel cells, they can recognize certain pathogen-associated molecular patterns. They'll have receptors for pathogen-associated molecular patterns, and these receptors on the surface of the sentinel cells are collectively known as pattern recognition receptors, PRRs. Okay, so this stands for pattern recognition receptors. So sentinel cells are covered in different receptors that are all clustered together under this umbrella term, pattern recognition receptors, and these are just receptors on the surface of the cell which have as their ligand these pathogen-associated molecular patterns. And of course, different pathogen-associated molecular patterns will have a different pattern recognition receptor. Okay, so the presence of the pathogen then will mean that there are certain pathogen-associated molecular patterns that are going to be exposed or are going to be displayed to our sentinel cells, and these will activate pattern recognition receptors on the surface of the sentinel cells, and then the sentinel cells will be activated to initiate the inflammatory response. And how do they do this? Well, they send out very powerful molecules that are going to act on the blood vessels, because the inflammatory response is all about changes in the microvasculature. So the sentinel cells will release molecules which we can collectively refer to as pro-inflammatory cytokines. I'm not going to go into the specifics of specific names for pro-inflammatory cytokines, but I'm sure you have heard of them before. Things like tumor necrosis factor alpha, interleukin-1, things like that, prostaglandin I2, all of these are clustered under this heading of pro-inflammatory cytokines. Now hopefully pro-inflammatory is pretty obvious. It's going to be in favour of the inflammatory response. Everyone always gets confused about this word cytokine. What is a cytokine? A cytokine is a really simple concept. It's just a molecule, here's the definition, it's just a molecule that is released by a cell of the immune system, so it's being released by one of these sentinel cells, that's a cell of the immune system, and it will act on another cell not necessarily a cell of the immune system now, the target doesn't have to be a cell of the immune system, and it will change the behaviour of that other cell. So a cytokine is effectively just a hormone released by an immune cell. It's just a molecule that's going to be released by an immune cell and which will affect the behaviour of other cells that aren't necessarily immune system cells. Okay, so the sentinel cells, once they've recognise the presence of the pathogen, they release pro-inflammatory cytokines, and there are loads of different examples of these, so we're just clustering them under this umbrella term of pro-inflammatory cytokines, and these are going to go and work on the microvasculature. They're going to work on the capillaries and the post-capillary venules and also the arterioles. So I just draw a little picture of this. So let's say we've got our microvasculature here, so here is our tiny little arteriole, which will now be splitting into tiny little capillaries here. So here's our capillary bed, and then of course they'll be reconverging up into a tiny little venule, like so. Okay, so let's just label this up. So we've got our tiny little arteriole here, which is bringing the arterial blood in, so I'll colour it in red here. Then we've got our capillaries in the middle here, so this is the capillaries. I'll colour those in yellow. So here are the capillaries, like so. And then we will have them reconverging up to form our tiny little venule over here. Okay, so this is the microvasculature in our tissue, and of course we'll have loads of these. We'll have loads of little arterioles splitting into capillary beds and then reconverging into venules. Okay, so the pro-inflammatory cytokines, they're going to work on the blood vessels, the microvasculature. So what are they going to do? Well, they're going to firstly act on the arterioles, okay, and they're going to cause arteriolar vasodilatation. So arterioles, they do have smooth muscle cells around them. They consist of endothelial cells lining the actual blood vessels sitting on the basement membrane, but then they do have a layer of smooth muscle cells around them. So they can change their diameter and change the amount of blood that's flowing through this microvasculature system here. So the pro-inflammatory cytokines cause arteriolar dilatation. They cause relaxation of the vascular smooth muscle cells. They don't quite do it directly. They act on the endothelial cells, and then the endothelial cells release something that makes the vascular smooth muscle cells relax. But overall, the effect is that we're going to get dilatation of the arterioles and therefore increased blood flow. So that's one of the components of the inflammatory response here. So blood flow is going to go up. Okay, so blood flow to the tissue is going to go up. That's first component of the inflammatory response. Okay, next, you're also going to get the pro-inflammatory cytokines acting on the endothelial cells of the capillaries and the venules here. Okay, and capillaries and venules, they have a very simple wall structure. They just consist of an endothelium, and that's it. They have endothelial cells sitting on basement membranes, so a very thin wall, the vessel wall here. And what's going to happen is they're going to cause endothelial contraction. So this is one of the things that's going to happen. You're going to get endothelial contraction. So what does endothelial contraction mean? Well, let me draw a little picture for this. So let's say this is the capillary or venule wall. So here is the basement membrane. So I'll just colour this in. So we're doing endothelial contraction. Here's the basement membrane in green here. And then we've got endothelial cells sitting on the surface here. So here's one endothelial cell, and here is another endothelial cell. Now usually they're nice and tightly together. The boundary between the two endothelial cells here is sealed. Okay, there's not a viable gap in between there, and not much of the blood plasma can actually leave the capillary and go into the interstitial fluid. What's going to happen when the endothelial cells get stimulated by the pro-inflammatory cytokines is that they're going to change shape. This is what it's going to look like after they've contracted down. So in a way very similar to the way muscle cells do, the endothelial cells are going to contract down like so. They're going to recoil their peripheral processes, if you like, and they're going to open up gaps in between the endothelial cells like so. So now you can see that there's a much bigger gap in between the endothelial cells. So this is the process of endothelial cell contraction, and this occurs not just in the capillaries but also the post-capillary venules. So you open up lots of gaps between the endothelial cells, and what does this now result in? So what does endothelial contraction produce? So this is going to result in the formation of an inflammatory exudate. So now that you've got these gaps in between the endothelial cells, fluid from the blood can leave the bloodstream and go into the extracellular space, the interstitial fluid, and you're going to therefore get accumulation of fluid there, and this new fluid that you're producing there is known as the inflammatory exudate. So this will cause the swelling of the inflamed area. So, just to go over this, increased blood flow is going to make the area red, it's also going to make the area hot. The endothelial contraction and the formation of an inflammatory exudate, that's what's going to cause the swelling of an inflamed area. So you form an inflammatory exudate. Now what's the point of forming this inflammatory exudate? Well, the point of it is that in the inflammatory exudate, and indeed initially starting at the first point, in the blood plasma, you have lots of proteins that are very good at attacking pathogens. So for instance, the complement proteins are the classical ones. These are fantastic at attacking pathogens, and they can actually kill pathogens. So the idea of forming an inflammatory exudate is that you're bringing all of these proteins that you've got within the blood plasma into the area where you've got the pathogen present, and they will then be able to attack the pathogen. So the point of forming an inflammatory exudate is to bring in lots of proteins from the blood plasma that are capable of attacking pathogens, and the sort of archetypal one to know is the complement proteins are in the blood plasma, and they're capable of attacking pathogens. Okay, so that's the purpose of forming an inflammatory exudate, and indeed this is why we want blood flow to increase to the area so that we can form more inflammatory exudate. So both of these work together to produce the inflammatory exudate, which is going to help us get rid of these pathogens. In addition, we're also going to get leukocyte recruitment. So this is bringing in proteins from the blood. Of course, we also want to bring in white blood cells. So leukocyte just means white blood cell. It's the fancy term for a white blood cell. So we're going to recruit white blood cells from the bloodstream. Now, of course, there are a lot of different white blood cells in the bloodstream. In the inflammatory response, we're only going to recruit very basic, blunt white blood cells. We're not going to recruit the fancy ones. They're part of the adaptive immune system, which we'll come on to in a moment. So the major leukocytes that are recruited in the innate immune inflammatory response are the phagocytes. You're going to recruit neutrophils and macrophages. So the two that you're going to have coming in are neutrophils. Majorly, they'll come in very quickly, so the moment the inflammatory response starts, you'll start getting neutrophils coming in, and then later on you'll start getting macrophages coming in. And they're both phagocytes. So if I just draw a picture of one of these, so let's draw a picture of a macrophage. So here is macrophage, so here's its nucleus here, and what they're capable of doing, both of these cell types, is they're capable of phagocytosing pathogens. They're capable of engulfing pathogens into a phagosome. So what can happen is they can engulf the pathogen. So here it is, our little red bacterium here. They're capable of engulfing it into a phagosome and then disintegrating it, digesting it. Again, that obviously clears the pathogen. So they're capable of phagocytosis, is the fancy word for engulfing pathogens and breaking them down. So you bring in the phagocytes, the neutrophils and the macrophages, which will start phagocytosing the pathogen. And this is a change that the endothelial cells have to make. So we've talked about how the endothelial cells will contract down and open up gaps in between them. These gaps are not big enough for leukocytes, such as neutrophils and macrophages, to fit through on their own. In order for a white blood cell to leave the bloodstream and go into the interstitial fluid, it has to get a lot of help from the endothelial cells. The endothelial cells have to have all sorts of molecules on their surface that interact with molecules on the surface of the leukocyte to negotiate the leukocyte's way through the gap in between the endothelial cells. And it is the pro-inflammatory cytokines which encourage the endothelial cells to actually start moving white blood cells from the bloodstream into the interstitial fluid. It does not just happen because of endothelial cell contraction. So this and this are separate things that the pro-inflammatory cytokines are going to produce. So overall, to summarize then the inflammatory response, the sentinel cells started off when they recognize the presence of the pathogen. They release pro-inflammatory cytokines which are going to cause vasodilatation of the arterioles to increase blood flow to the affected area, and we're going to get endothelial cell contraction of the endothelial cells in the capillaries and the venules, which is going to allow us to form both an inflammatory exudate, which will have all sorts of proteins in it that can attack the pathogen, and also the endothelial cells are going to be encouraged to start recruiting leukocytes into the interstitial fluid, so you'll start getting neutrophils and macrophages being moved from the bloodstream into the interstitial fluid, and they will start phagocytosing the pathogen. And that's the inflammatory response. That is the major way in which the innate immune system deals with an infection in a tissue. Okay, right. Now, in order to maintain the inflammatory response, you have to continuously produce more pro-inflammatory cytokines. So whilst the pathogen is still present in the area, the sentinel cells and also a lot of the other cells that you've brought in, so for instance all of these white blood cells that are coming in, they will start continuously producing the pro-inflammatory cytokines and continuously maintaining the inflammatory response, maintaining this activity on the microvasculature, and until the pathogen is actually being cleared, and then the pro-inflammatory cytokine level will fall. So it's important to remember that pro-inflammatory cytokines are continuously being produced in order to maintain the inflammatory process going on. If they were to stop being produced, then the inflammatory response would die. Okay, right. So, an anti-inflammatory drug or an anti-inflammatory effect, when people talk about a molecule having an anti-inflammatory effect, for instance cortisol has an anti-inflammatory effect, hopefully you are now able to have a good guess at what that means. It means that it's going to reduce the inflammatory response. Okay, immunosuppression, which we'll come on to, is all about reducing the adaptive immune system. So anti-inflammatory means we're going to be inhibiting the innate immune system. Immunosuppressive means that we're going to be inhibiting the adaptive immune system, which we'll come on to in just a moment. Okay, so cortisol has both an anti-inflammatory and an immunosuppressive effect. We'll come on to its immunosuppressive effect once we've discussed the adaptive immune system, but let's for now talk about its anti-inflammatory effect. So how does cortisol reduce the inflammatory response all over the body? Well, it goes into all sorts of cells. We've discussed that cortisol is lipid soluble, so it can get into pretty much any cell you can imagine. So it goes all over the body and is going to affect tissues all over the body. And indeed, it goes into these sentinel cells, for instance, and the change in behaviour that it will produce is it will reduce the release of pro-inflammatory cytokines. It can also go into the white blood cells, which I've said later on, to maintain the inflammatory response, will be releasing pro-inflammatory cytokines as well, as long as the pathogen is still present. So if the neutrophils and the macrophages are still busy phagocytosing pathogen, then they're clearly aware that there is still pathogen present in this area, and they release pro-inflammatory cytokines to maintain the inflammatory response. They're saying, we've still got lots of work to do here, keep bringing in more inflammatory exudate, bring in more cells. So cortisol will also go inside these cells and reduce their release of pro-inflammatory cytokines. So this is the way then that cortisol reduces, dampens down the inflammatory response. And we'll talk about this more later, but why would you want to do that, you might ask? Well, it's to dampen it down. I mean, biologically, it doesn't completely stop inflammation in the physiologically relevant levels that it's present at. What it does is it dampens down the inflammatory response, and if you have too little cortisol, what you find is that you get far too large inflammatory responses that cause massive tissue damage. So it seems as though it just puts the brake on the inflammatory response and keeps it at a safe level, because this response could be extremely dangerous happening in tissues. It causes huge damage to the tissues. I know the pathogen's present that needs to be annihilated, but the actual inflammatory response is going to cause major tissue disturbance and damage as well. So we want it to be controlled, we don't want it to be too large, and cortisol seems to be one of those things that puts the brake on the inflammatory response and makes sure that it doesn't go out of control. So in people who have far too little cortisol, they have far too large inflammatory responses generally, which can cause a lot of tissue damage. Okay, so cortisol is going to have an anti-inflammatory effect by going into all sorts of cells and stopping the production of pro-inflammatory cytokines, or at least reducing it down, and that therefore reduces the inflammatory response. It reduces its initiation and also reduces its maintenance. Okay, so that's cortisol's anti-inflammatory effect. Let's now move on to its immunosuppressive effect. So as I say, anti-inflammatory, when someone talks about an anti-inflammatory effect, it means we're dampening down the innate immune system. When we talk about immunosuppressive drugs and molecules having an immunosuppressive effect, this means that we are dampening down the adaptive immune system. Okay, so let me just remind you then of the adaptive immune system and what this involves. Okay, so we haven't really got much space here, so I think I'll go over onto the other side to discuss this. So the adaptive immune system is very complicated, but we can, for simplicity, divide it up into just two different branches of the adaptive immune system. So I'll just put this here. So the adaptive immune system is what we're discussing. Okay, so the two different branches of the adaptive immune system are the humoral response of the adaptive immune system, and then the cell-mediated response of the adaptive immune system. Now, what's the difference between these two different parts of the adaptive immune system? And they're very different responses. Well, the thing is, they respond to different types of pathogen. The humoral adaptive immune response is there to respond to extracellular pathogens, whereas the cell-mediated response is more about responding to intracellular pathogens. So let me just write that down. So the humoral adaptive immune response is the adaptive immune response that we would use to destroy extracellular pathogens, and the cell-mediated adaptive immune response is the one that we'd use to destroy intracellular pathogens. Okay, so what do I mean by an extracellular and intracellular pathogen? Well, let me just spell this out. An extracellular pathogen, for instance, is the sort of pathogen we've been talking about at the moment. It's a bacterial cell, for instance, would be the classical example, that is just sitting beside your cells. So if I draw a picture here, let's say these are the normal human cells. So this is a human cell, this is a human cell, this is a human cell, and then we've got the pathogenic cell, and it would generally be far smaller than the human cell. But just for the basis of the picture, let's say here is our bacterial cell, and you can see that the pathogen is sitting in the interstitial fluid. It is not inside the actual cells themselves. It's just amongst your cells, and we don't like the fact that it's there. So that is what I mean by an extracellular pathogen. An intracellular pathogen, on the other hand, is where the pathogen is actually inside your cells. So now let's draw a picture of a human cell. So I've zoomed in a bit now, and we've got the pathogen inside the human cells. So for instance, a classical example of a pathogen that will go inside of cells is a virus. So let's say this is the nuclear capsid of the virus. There's maybe its genome. Okay, so viruses are a classical example of an intracellular pathogen. So this might be a virus. But, you know, other pathogens can also be intracellular pathogens. So, for instance, tuberculosis, Mycobacterium tuberculosis, it classically goes inside of cells. Lots of the fungal cells also invade your cells, and they're parasites inside your cells. Malaria goes inside your cells. So there's loads of different pathogens that can all go inside of human cells. So we can divide pathogens into extracellular pathogens and intracellular pathogens, and the adaptive immune response to these two different types of pathogens is going to be different. So we're going to have the humoral adaptive immune response for extracellular pathogens and the cell-mediated adaptive immune response for intracellular pathogens. So let me very briefly outline what these two immune responses involve, and I could talk for hours about the humoral and cell-mediated adaptive immune response. I'm not going to, I'm just going to give you the broad outline. So in the humoral, and this is oversimplified, but it's a simplification that's okay to use, and it keeps it very simple in your mind. Okay, so basically for the humoral adaptive immune response, what happens is initially you activate CD4 positive or helper T cells. So in the humoral adaptive immune response, what you get activated is CD4 positive, which are also known as helper T cells. Okay? And these CD4 positive or helper T cells are going to be specific against the pathogen. So they will be targeted specifically against some sort of antigen that pathogen has present on it, or as part of it. So these are specific to the pathogen. You have loads of different types of T cells, and they are all targeted against different pathogenic antigens. Okay, so as part of the humoral adaptive immune response, you'll get CD4 positive helper T cells activated, and these are then going to activate B cells, which again will be specific to your pathogen, and the B cells will then become plasma cells and produce antibody molecules against your pathogen. And these antibody molecules will then be released into the bloodstream, and they will go and bind to the pathogen, bind to the specific molecule on the pathogen that they're targeted against, and they'll aid in the destruction of that pathogen. Okay, so that in brief then is what the humoral adaptive immune response consists of. You activate helper T cells, which are also more fancily known as CD4 positive T cells, which then are involved in activating B cells, which are directed against your pathogen, and then the B cells will produce antibodies that are specifically targeted against your pathogen. Okay, so that's the humoral adaptive immune response. In the cell-mediated adaptive immune response, you activate CD8 positive or cytotoxic T cells. So this involves CD8 positive, or you can also call them cytotoxic T cells. And these again will be targeted specifically against this pathogen. Okay, so they are specific to the pathogen. And what they will then go and do is they have the power to actually kill cells, human cells, that are infected with the intracellular pathogen. So these are very powerful cells. They have the ability not just to kill pathogens, but to kill cells that are infected with pathogens. So you activate these CD8 positive cytotoxic T cells, and these will go round, and they have the ability to recognize which cells of the body are infected with the pathogen that they're targeted against, and they'll find these cells and they will kill them. So that's what the cell-mediated adaptive immune response involves. So that's a very sort of brief outline of, ridiculously brief outline of the adaptive immune response. So just to summarize again, the adaptive immune response has these two very separate arms: the humoral adaptive immune response and the cell-mediated adaptive immune response. The humoral is for extracellular pathogens and involves activating CD4 positive, which you can also know as helper T cells, which are specifically directed against that pathogen. And they will then activate B cells that are specifically directed against your pathogen, and the B cells will then produce antibodies which are going to be targeted against your pathogen, and they will be released into the blood, go and find the pathogens and result in their destruction. Okay. Meanwhile, in the cell-mediated response, and actually it might just be good if I explain why this would be no use against an intracellular pathogen. This is only useful against an extracellular pathogen because antibody molecules cannot get inside of cells. So if you've got an intracellular pathogen, there's absolutely no point filling the blood with antibodies against that pathogen. It's just not going to work. I mean, obviously the pathogen might occasionally be extracellularly, and the antibodies will help to deal with that, but it won't get rid of the pathogen that's intracellularly. So that's why we need a separate response for the intracellular pathogens, and this involves activating CD8 positive or cytotoxic T cells, which are very powerful cells, very scary cells. These will be directed specifically against the pathogen, and they will have the ability to go round all over the body and look for cells that are infected with this pathogen that they are directed against. And if they find a cell that is infected with this pathogen, then they will destroy that cell, and obviously they'll destroy the pathogen that's inside it as well by destroying the cell. Okay, so that's the adaptive immune system. It's also worth explaining that to keep the adaptive immune system running, you have to continuously be reinitiating the activation process. So when the pathogen is present, you will activate a whole bunch of CD4 positive helper T cells, which will then activate those of B cells, which will then produce lots of antibody. But these cells don't last forever. You have to continuously be doing the process over and over again. These cells will be cleared from the body within a certain period. So you'll then be reactivating new CD4 positive or helper T cells that are specifically directed against the pathogen, which will then be activating new B cells, which will then be producing antibody. And the same for the cell-mediated response. You're continuously going to have to be activating more CD8 positive cytotoxic T cells that are directed against the pathogen. Okay, so to maintain the adaptive immune system on, you have to continuously be activating T cells is the common theme here. Now, cortisol achieves an immunosuppressive effect, i.e. it turns these two branches of the adaptive immune system off, or at least tones them down, by going inside of CD4 positive and CD8 positive T cells and preventing their activation. Okay, so it prevents activation of T cells. So prevents activation of both types of T cells, both CD4 positive, the helper, and CD8 positive cytotoxic T cells. Prevents activation of T cells, because the pathway by which they activate is very similar, well, it's the same in both of them pretty much, and cortisol is going to target the same part of that. Okay, so it prevents the activation of T cells, and therefore it's going to prevent, or at least tone down, this maintenance of the adaptive immune response. So as I say, to maintain an adaptive immune response, you have to continuously be reactivating more T cells and continuously be producing new adaptive immune system cells that are going to keep going, because you don't keep these cells that you've produced forever. They are gradually removed from the bloodstream. They don't live forever. So if you're preventing the activation of T cells, then you're going to be stopping, or at least reducing, the amount of new cells that you're producing, and therefore your old ones will gradually be cleared, so gradually your adaptive immune response will go down. Okay, and again, why would you want to be doing this? Well, again, cortisol is producing a brake on the adaptive immune response. The adaptive immune response is potentially, just like the inflammatory response, extremely dangerous, extremely damaging to your own tissues, and therefore we have a brake to keep it under control. And people with far too little cortisol, not only do they get excessive inflammatory responses, but they can also get excessive adaptive immune responses. Okay, right, so that's the immunosuppressive effect then of cortisol. So we'll have a break here, and in the next video what we'll actually talk about is the control of the synthesis and secretion of cortisol by the hypothalamic-pituitary-adrenal axis.